is N2. Elements with protoplasm and proteinaceous material N2. Amino acid has hydroxyl in its R group, serine. Excess carbohydrates and proteins stored in body as fats. Charge group of opposite polarity, zwitter ion. Nucleotides, purines, pyrimidines, sugar, phosphate. 98% are living organic, carbon, hydrogen, nitrogen, oxygen, phosphorus and sulfur. CH3, CH2, 14, COOH, palmitic acid. Triglyceraldehyde, fat stored in form of droplets. Membrane lipid, phospholipids. Steroids, animal hormone. Wax, feather and leaves. Essential fatty acids, arachidonic acid. Secondary metabolites and tables in NCERT देखना है और पढ़ना है, बहुत अच्छे से याद रखना है. Secondary metabolite is a drug, vinblastine. Lectin, convalin, proteins, heteropolymers containing strings of amino acid. Arbin, toxin. GALT4 enables glucose transport into cell. Collagen, intercellular ground substances. Convalin, lectin. Most abundant protein in animal world is collagen. Fish more nutritionally क्योंकि egg 20 amino acid rich in. Collagen, intercellular ground substances. Trypsin, enzyme, proteinaceous. Protein nature. Insulin, hormone. GALT4, glucose transport. Most abundant protein living world, Rubisco of plant and collagen of animal. Protein, heteropolymer, amino acid made of. Ribozymes, nucleic acid plus catalytic power. 3 types of macromolecule found in living: protein, nucleic acid and polysaccharide. Amino acid names पूरे 20 के. सेल्यूलोजिक अमीनो एसिड के नेम्स देखो अपनी NCERT से और मॉड्यूल्स वो जो सर ने करवाए थे ना, ST अल्कोहल और हेज़वाइफ एरोमेटिक, वो वाले पूरे. सेल्यूलोज होमोपॉलीमर. 1 degree metabolic ribose. Secondary metabolite anthocyanin. Protein, insulin. Enzyme, all proteins, highly specific. Amino acid linked by peptide bond. Glycogen, homopolysaccharide, branch chain molecules stored in liver and muscles. Chitin is macromolecule, not protein, a polysaccharide, and 2 contain polysaccharides. Cellulose is a polymer, beta-D glucose. Insulin is a polymer of fructose, homopolysaccharide. Monomer unit of chitin and fungus cellulose and acetyl glucosamine. Whole biosphere most abundant Rubisco. Structural polysaccharide, chitin. Polysaccharide plus beta glycosidic bond is cellulose. Polysaccharide, glucose. Homopolysaccharide is starch, insulin. Ribose, monosaccharide. Starch make it useful to store material. Chemically non-reactive and osmotic inactive. Chemically non-reactive and osmotically inactive. Starch in polymer of glucose, disaccharide, lactose, sucrose, maltose. Monobasic amide, starch. Polysaccharide, cellulose. Thylakoid, stacks of coins from granum. Majority of enzyme contains non-protein prosthetic group. 3,7 polyhydroxylactone monosaccharide. Maltose, polymer of ग्लूकोज होगा ना, माल्टोज पॉलीमर, उसका तो लिखा ही नहीं है. फूड ट्रांसपोर्ट इन प्लांट्स, सुक्रोज. नॉन रिड्यूसिंग कार्बोहाइड्रेट, सुक्रोज. माल्टोज का पॉलीमर क्या है? एडेनाइन थाइमाइन प्यूरिन्स. Ribose sugar occurs in RNA and ATP. Adenylic acid. Nucleic acid double ring formed in guanine. Cytosine. Anarchidoic acid molecule C. Palmitic C. ये कुछ पॉलीपेप्टाइड aquaporin, uracil, nitrogen base uracil, nucleoside cytidine, nucleotide thiamidylic acid, nucleic acid RNA, adenylic acid nucleotide, uridine present only in RNA is nucleoside, nitrogen base plus pentose sugar plus phosphate group nucleotide. Nucleotide is uracil. Phosphodiester bond nucleotides in nucleic acid. Ribose of RNA OH group in second position. Stretch of DNA consisting of 10 to 20 base polynucleotide. Polypeptide subunit present in adult hemoglobin. 2 alpha and 2 beta subunits. Structure function as enzyme is tertiary. Not all proteins have quaternary structure. Secondary structure helix and alpha helix and beta pleated C. Basic amino acid C NH2 H COOH CH2 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 NH2. Quaternary structure immunoglobulins. Quaternary structure hemoglobin. Quaternary structure dictated by 1 degree primary structure of individual subunits. Not common in protein quaternary structure. Amino acid के structures identification NH2 CH COO ग्लूटामिक एसिड. आइडेंटिफाई करो अमीनो एसिड्स को पूरे जो सरने में थे इज वाइफ एरोमेटिक एंड STM. ग्लूटामिक एसिड का स्ट्रक्चर दिया है यहाँ NH2CH और CO2 CH2CH2COOH. नाइट्रोजन बेस लिंक टू शुगर थ्रू ग्लाइकोसिडिक बॉन्ड. Structure 1 degree peptidal stabilizes or secondary starch peptide maintained by hydrogen bond. ATP 0 phosphodiester bond. Polysaccharide monomers linked by glycosidic bond. Formation of peptide and glycosidic bond dehydration. Formation of peptide bond involves 2 amino acids and a condensation reaction. Nitrogen base is linked to sugar by a glycosidic bond at carbon number 1. Major and minor groups occurs in DNA. DNA can be A, B, Z DNA. Bond between 2 carbohydrate molecule glycoside. High energy bond ATP PO. ATP nucleotide. Decreases Km value, increases affinity with substrate. Oxidoreductase dehydrogenase. Isomerase interconversion of isomers. Ligase is link of 2 compounds. Lysase removal of group from substrate. Nomenclature of enzyme is done on basis the type of reaction catalyzes. Coenzyme NAD+. Coenzyme NAD and NADP contains vitamin niacin. The essential chemical component of many coenzymes are vitamins. Transition state structure transient and unstable. Prosthetic group non protein metal compound tightly bound in enzymes. X is temperature, Y is enzyme activity. Sugars act split further glucose. Apoenzyme protein. Thermolabials protein part enzyme. Thermolabials मतलब unstable. Apoenzyme. Holoenzymes. Apoenzyme plus coenzyme SZ plus S dash gives S plus S dash plus S dash G. Transferase. Heme act as prosthetic, catalog and peroxidase. Coenzyme and prosthetic group both requires for enzyme action. Both are organic compound. Both are not a polypeptide. Not true about coenzyme. Both are separate from enzyme. Both are separate from enzymes. Not true about coenzymes. Both are separate from enzymes. Cofactors of carbonic anhydrase zinc. Class of enzyme in lysosome hydrolase. All enzymes are protein except ribozyme. Activity of ribozyme is affected least by original activation energy of system. Metal ion requires for normal functioning of enzyme cofactor. Digestive enzymes are hydrolase. Not a protein, ribozyme. Organic substance bound to an enzyme and essential for its activity is coenzyme. Non-protein part of enzyme is cofactor. NAD, nicotinamide adenine dinucleotide. A is normal enzyme activation. B is competitive inclusion. C is non-competitive inhibition. Enzyme catalyze removal of a group and formation of double bond. Lysis. Enzyme catalyze removal of group and formation of double bond. Lysis. Holoenzyme combine coenzyme and apoenzyme. Molecules function as biocatalysts in addition to enzyme RNA. Triple helix structure of collagen. J.N. Ramachandran. Prosthetic group of glycogen consists of carbohydrates. Lactose, galactose. RNase, uracil. Beta pleats, secondary structure. B wax, palmitic acid.